Hi, I'm Dr. Asegun Henry, and I'm a professor in mechanical engineering at MIT. This is the third episode of a new YouTube series I created called Ask a Real Scientist. This series is about educating the general public about science, technology, and engineering, and specifically increasing scientific literacy. This episode is a follow-up to the first episode, which is about COVID-19. Here, I'm interviewing another colleague, Dr. Angela Rasmussen, who's a virologist at Columbia University. This episode specifically deals with this question of whether or not you can get the SARS coronavirus 2 more than once. And in this episode, we also provide a general overview about how vaccinations work and also how testing works. I think you'll find her perspective very enlightening.
So I'm Angela Rasmussen. I'm a virologist at Columbia Mailman School of Public Health. I study the host response to virus infection. And what that means is that most people know when you are infected with a virus or your cell is infected with a virus, that virus hijacks the host in order to replicate itself. And that's basically what viruses do. The host can recognize that there's a virus there, and it mounts a response. And sometimes the interactions between the virus and the host, as well as that host response, can determine whether or not the virus goes away, gets cleared, or actually makes the disease worse. So I study that for emerging infectious diseases, exclusively viruses. I've worked on avian influenza. I've worked on MERS coronavirus. I have largely worked on Ebola virus for the last few years. And now I am also doing that type of work with SARS coronavirus too.
I think one of the big questions that's on, I know, a lot of people's minds is really about this notion of can you get it twice or can you get it 3 times, or and I guess can you speak to mechanistically how immunology works? What you just talked about, a virus gets in, hijacks the cell. Can you talk a little bit more about different viruses, how they behave, and like why are there some viruses that you can get more than once versus others that you don't, and how does all that work?
Sure. So there are really 2 components at play here. And again, this is really the central part of my research question, and that's the relationship between the virus and the host. So there are some viruses that you can get again, although in those cases, you're not really getting the same exact virus. So take influenza, for example. Most people know that we have to get a flu shot every year because the flu virus changes. And that is because the flu virus, which are organized into what we call subtypes, so that's H1N1, H5N1, all of those HN numbers that many people are familiar with, those are all different to your immune system. So within each of those categories, the viruses can be different from each other. That's like here in Seattle, there's a lot of people, we're all people that live in Seattle, but we're inherently different from one another. And your immune system can't necessarily recognize that you are the same as me, and that we're both the same species. It will recognize us as different entities. And so that's the situation with influenza virus. Other viruses are much more stable, meaning that there's really only one kind. So that would be like a population of people who are all clones of each other, or very, very similar to the point that you couldn't really tell them apart. And that's how the immune system distinguishes between different viruses. That's why you have to get a flu shot every year to protect against the new strains of flu that are emerging. One thing about SARS coronavirus 2 that we don't know is its ability to form these immunologically different strains. So far, it doesn't appear that it is changing to the point that immunity to the SARS coronavirus 2 strains that are circulating now will not protect you against the ones in the future. But really the only way we can tell that, because this is a novel virus, is to keep following the virus and see if it changes, if it does evolve that ability to be not recognized by immune responses to the virus that's circulating now. There's the other factor, so the immune system. When you are infected with a virus, your body mounts an immune response to it. Typically, a consequence of this immune response is that you develop antibodies and immunological memory, meaning that you have these antibodies, which are essentially, they're little Y-shaped proteins that circulate around in your blood all the time. and they bind that virus very specifically. So if you think of viruses as a population of people and you get infected with the antivirus, you will have antibodies that recognize exclusively me. They would not recognize you. And so that's how vaccines work. That's how the immune system works in most cases. There are, of course, exceptions. One example of a case where you can get a virus twice, but it's not really getting a virus twice, it's having a virus become reactivated, is herpes viruses. So chickenpox, for example. If most people are familiar with chickenpox, you get it most of the time in childhood. It's a pox-like rash all over your body, and then it goes away, and that's because your immune system clears it out. But those viruses have the ability to hide in cells that are protected from the immune system. So your neurons, you don't want your immune system to go around killing them because we can't replace them. So herpes viruses like chickenpox hide out in those nervous tissue cells and neurons, and it just sits there dormant for years. Then it could be decades later, you have some kind of stress, something that causes that virus to reactivate. It turns all of its genes back on, and you get the virus again, except this time it's in a different form called shingles. And that is largely, we don't really understand how the immune system controls that during that, what we call the latent period between chickenpox and shingles. But that is one scenario in which the immune system does control the original infection, but it sticks around and kind of hides out from the immune system. We've evolved, and viruses have evolved, a number of different ways to keep trying to one-up each other, essentially, in this sort of constant struggle between the virus and the host. And I like to think about it as almost like a football game that never ends and has an infinite number of plays in the playbook. It really is, viruses and hosts are constantly evolving and adapting to each other. In this situation with SARS coronavirus 2, one important point that I think often gets lost is that we haven't actually been evolving with this virus for a very long time. It recently emerged into human hosts. So this virus is adapted for bats. And sometimes with these novel emerging viruses, one thing that happens is because they're completely new to us and our immune systems, the virus hasn't adapted to persist in us, and we don't necessarily have great strategies from an evolutionary perspective to deal with it, which is why we may be seeing so many different complex disease presentations with this.
So it sounds like it in part comes down to diversity. So for a given pathogen class, I guess, there's a certain amount of diversity within it. And if you have a particular class that's not very diverse, then there's a high likelihood that human beings can build some immunity and keep this memory you were saying, and be able to fight off those small variants that might take place. But if there's lots of diversity, then it can be a really big, difficult battle, because any significant variation that happens now requires a new immune response, and you have to expand the memory to develop more. So
that's absolutely correct.
Because I think the main question a lot of people have is, if they get it, and let's say they recover from it, now we're talking like 4 weeks later, are they safe to go out now? Because if they get exposed to it again, they should. I mean, and one way to take what you're saying, it seems like they should have the memory from 4 weeks ago and maybe still be able to manage it. I don't know, do viruses mutate and evolve that quickly? In 4 weeks, there could be some modified version that now they don't have the immunity to?
So that's an excellent question. And part of it has to do with the fact that coronaviruses are RNA viruses. So their genome is made of a molecule called RNA, which is very similar to DNA, but it's different in one very important way, and that is that it's less stable and it's more prone to mutation. So the enzymes that actually replicate the genetic code of an RNA virus can't correct any mistakes they make when they're copying that genome. So RNA viruses in general have a much higher mutation rate than DNA viruses or organisms like us that have DNA genomes. And that's because the DNA enzymes, they're called polymerases, have proofreading capabilities, whereas RNA polymerases do not. However, coronaviruses have another enzyme that has some partial proofreading capability. So coronaviruses have a lower mutation rate than other RNA viruses, even though that's still higher than DNA viruses. So what that means is that coronaviruses do mutate. They do acquire mutations every time they replicate, but they don't do it as fast as other viruses. So that does make it less likely that this virus will rapidly evolve escape mechanisms the way that we say influenza could. That said, we really just don't know enough about immunity. So there's really two factors with regards to long-term immunity. One is the virus and how diverse is that virus, as you correctly observed. The other is how long does that immunological memory last and how long is it functional? So theoretically, we should get infected once and never be able to get infected again. And many of our most successful vaccines work that way. So the polio, the oral polio vaccine, for example, old people like me, got the oral vaccine, and I got that once when I was 2, and I'm presumably protected for life from polio. I had my last MMR shot when I was, I think, in high school, and I just had my measles titers taken because of vaccine denial and the increase in measles cases. I was traveling abroad last fall, and so my doctor recommended that I have my antibody levels checked for measles antibodies, just in case I needed a booster of my MMR shot. And I still had high titers of antibodies to measles in my blood from that vaccine that I received, over 20 years ago. So many vaccines and many viruses do induce robust, lifelong memory immune responses, but some viruses don't. And that's a very active area of study. What we don't know about SARS coronavirus 2 is how long, actually, if it produces functional immunity, although studies now seem to suggest that yes, people who are infected do have antibodies, and that those antibodies are what we call neutralizing, which means that they can render the virus non-infectious. So they block the virus basically from infecting other cells, and they essentially latch on to the virus and keep it from being able to infect anything. We do think that most people develop neutralizing antibodies to SARS coronavirus 2. But what we don't know, because this virus just emerged 5 months ago, is how long those neutralizing antibodies last and how high of levels of those neutralizing antibodies you need to have in your blood in order to be protected. So that is all research that's going on right now. And obviously that has important implications for developing a vaccine, because what vaccines do is they basically trick your body into making these immune responses without having to get infected with the actual virus. So one really important question for the vaccines that are in development right now is, do they first of all induce protective immunity? Do they induce these neutralizing antibodies? Are they inducing other cells of the immune system to react protectively and specifically to SARS coronavirus 2? And if so, how long does that protection last over time? Unfortunately, the only way you can tell how long it lasts over time is to follow people over time and see how long it lasts. So there's not really a way to speed up getting answers to that particular question. But the early data so far does suggest that people do develop immune responses, and those responses are probably protective.
How, in general, how does a vaccine work? There's a lot of fear around vaccines, and there's a lot of criticism over lack of information and possibly lack of safety around vaccines. And so, from what I understood that you just said, is essentially it seems like a vaccine is intended to stimulate your body's immune response to develop the antibodies that would fight the virus without being the virus itself.
Yeah. So that's how, that is how vaccines work. Vaccines work basically by fooling your immune system into thinking that you've been infected with the virus without having to actually be infected by it. And certainly we can develop immunity by getting infected to a virus. But it's a lot better, and certainly a lot less people die from it, if you don't have to infect anybody with the virus, but you can still elicit that immunity. And especially if you do it over a population, then we get what's called herd immunity, and not everybody has to be vaccinated because there are so many immune people walking around that a virus can't get a foothold in a population. And one important concept to understand with regards to herd immunity is that viruses are obligate parasites. So they can't replicate without a host. Viruses can't just go off and like live in a pond or, you know, in the grass or in the environment or in your house. They're not like bacteria where they're in the water dividing on their own. They have to have a host. So if you have most of the population immune, a virus can't spread from person to person because everybody is not susceptible to that virus, or the vast majority of people are not susceptible to that virus. The way that we would prefer to do that is not by everybody getting infected and developing immunity. It's by vaccinating everybody so that nobody has to actually get infected in the first place. And there are a number of different vaccine strategies that we've used over the years to do this. So there's the inactivated vaccine. And I guess before I get into this, let me just say that the basis for which the immune system specifically recognizes viruses is based on shapes. So if you think of a virion, a virus particle, as like a golf ball, covered with little dots, covered with little balls, for example. Most people know at this point, especially what a coronavirus looks like, because there's pictures all the time on the news of the coronavirus. Coronaviruses actually look like a crown because they have these spiky shaped proteins coming off of them. And that protein was cleverly named by virologists as spike. So spike is what your immune system will recognize, and it will recognize that based on the shape of those spikes. So the antibodies, if you think of them sort of like, you know, a hand, it's like a hand that only specifically can grasp an object that has a very specific shape. And that's the basis by which antibodies and T cells, actually, that's the basis by which your immune system recognizes whether something is foreign or not. Okay. So what you want to do when you're making a vaccine is to basically figure out a way to get those virus shapes into your body so that the immune system can recognize them and make a response. Now, the immune system is a marvel, and I won't get into all of this because it's incredibly complicated, but the immune system develops randomly antibodies to a huge assortment, on the order of potentially quadrillions or quintillions. So, you know, 3 orders of magnitude bigger than a billion. Different possible combinations for shape recognition. So you have all of these antibodies circulating at very low levels around. If one of those antibodies actually finds the shape that it normally goes with, that's what triggers the immune response. So if you get those shapes into a person, and those low-level antibodies and what we call naive B cells that secrete them, recognize that shape, that gives a signal to the cell to start dividing and start turning itself into a memory B cell that can make these antibodies in perpetuity, theoretically. So there's a number of different strategies for actually doing this. One of the easiest, I think, for people to understand is an inactivated virus. That's when you just take a bunch of the virus, you grow it up in cell culture, you inactivate it somehow while still preserving the shape. So you don't blow it up, you don't lyse it, which means that you basically disrupt it and dissociate the particles. You inactivate it in a way that preserves the shape of the virus particle. And then you put that into a person. And examples of vaccines like that are the flu shot. So the flu shot is an inactivated virus vaccine. The polio vaccine that people get now here in the United States is an inactivated vaccine as well. That's the Jonas Salk vaccine, for people who know about vaccine history. And so that's one example. Another example is a live attenuated virus vaccine. That's the Sabin oral polio vaccine. So this is when you take a virus and you do something to it to make it less pathogenic or less capable of causing disease. Then you infect people deliberately with that weakened virus. It doesn't cause disease in them. It replicates, it makes more of the viral proteins that the immune system is going to recognize, and triggers immunity that way. So in that case, you're actually getting infected with the virus, but it's not going to make you sick. There are new like second and third generation vaccine technologies as well. So there's DNA and RNA vaccines, and the SARS coronavirus mRNA vaccine made by Moderna that's currently in clinical trials is an example of this. That's when you take a piece of DNA or RNA that encodes the viral gene that you want to develop immunity to, and you put that into people. And so you trigger your own cells to make that protein. Your immune system recognizes it as foreign and mounts an immune response to it. And one of the advantages to that is you're not being infected or exposed to any kind of virus at all. You're just making a single virus protein, and that's relatively easy to manufacture those vaccines. There are some challenges with actually getting your body to respond immunologically to those vaccines. There's also subunit vaccines, which are basically giving you the protein the virus protein that your immune system is supposed to respond to. And then there's another type, and a good example of this is the recently approved Ebola vaccine called a viral vectored vaccine. And in that one, you take a different virus that doesn't cause disease in people, and you genetically engineer it so that it's expressing the protein that you want immunity to. And then you infect the person with that virus. That virus replicates. Usually they are engineered also so that they don't replicate too much. They can't grow and cause an infection really themselves. They just replicate enough so that they're producing this protein, they're stimulating the immune system, and you develop then an immune response to the protein from the virus that you're trying to vaccinate against. And that strategy is also being used for SARS coronavirus 2 by the University of Oxford. So all of these vaccine strategies are in play for SARS coronavirus 2. And I think, personally, I think it's really good to try all of them, because they all have their advantages and disadvantages in terms of both the type of immunity that they trigger. And really, that's the only way we figure that out is by testing them and figuring out which vaccine works best for this virus. But they also have different advantages and disadvantages in terms of scaling them up and distributing them to large populations, which is another huge issue for this particular virus, since this is such an urgent public health crisis.
I guess it makes sense now that you can have your body figure out how to deal with it without having such an extreme response that now can be all the symptoms. So
you can sort of think of vaccines almost as a controlled burn. And when you're infected with a virus, one thing we really don't understand actually about a lot of viruses is why one person gets really sick and one doesn't. But of course, there are side effects to vaccines, and
that's what I was going to ask about next. So, well, go ahead. Let me let you go.
Yeah, so that's why, I think it's incredibly important. I understand the urgent need for a vaccine for SARS coronavirus 2, but I'm very hesitant to say that we should completely chop off a lot of the normal parts of evaluating a vaccine. So we need to do these large randomized controlled clinical trials with these vaccines, because there have been examples of vaccines that failed in trials because they were not necessarily effective, but also that they were not safe. And some of those can be that the vaccine has unforeseen consequences, in that it can induce immunity to the virus, but also can trigger autoimmune diseases. So if the shape of a particular part of that viral protein that the immune system is recognizing is too much like a shape within one of your proteins within the human body, that can have really bad effects. And that means that the immune response that's mounted against the virus can cross-react with whatever protein is in your body, and this can have all types of consequences. Sometimes vaccines can elicit unforeseen immunological or inflammatory responses that can result in sort of long-term either autoimmune disease or chronic inflammatory disease. So one really critical part of the vaccine clinical trial process usually is first actually to develop it, test it in animals to make sure that it's likely to be safe in people. But absolutely a step that we can't skip is the phase 1 clinical trial in humans, which tests exclusively safety. That's just to see if you give a person this vaccine, are they going to get sick from the vaccine? And they are insisting on doing these phase 1 clinical trials for all of the vaccine candidates here in the US. So that certainly makes me rest a little easier knowing that there is going to be safety data before vaccine candidates move into the next phase, which is to actually test efficacy or whether or not that vaccine actually works. And normally this process takes years, and there's typically 3 phases of clinical trials where phase 1 is a safety trial, phase 2 is a safety and efficacy trial in a larger but not gigantic number of patients. Phase 3 is a large efficacy trial, usually involving hundreds or thousands of patients. And that usually can go for up, over a year, because again, to determine whether there's long-term protective immunity, you have to look over a long-term scale thing. And at the end of that period, we'll be able to look back and say the people who got the vaccine did not get infected with the virus. They did not ever develop COVID, and their antibodies appear to be neutralizing. That will be an example of a successful vaccine. If some of those people do get infected, or if they're associated with transmission, then we might be able to say, well, the vaccine works for some people, but not for others, or there's partial protection, but not complete protection, which is what we see oftentimes with the flu shot every year. So those efficacy trials are really important. Right. And the safety profile, to be clear, while there have been vaccines that failed clinical trials, either because they weren't safe or effective, those are not the vaccines that are available to us now. The vaccines that are available and approved for use in the clinic have excellent safety profiles. There's a very, very low rate of serious adverse effects to most vaccines that are available for people.
So every year there's a new flu vaccine. How is it that the flu vaccine gets approved so quickly?
So the flu vaccine is the same platform every year. It's the same method used to grow the inactivated flu viruses. And again, those are inactivated viruses. So you're not actually getting infected with anything when you get the flu vaccine. From a sort of pharmacological perspective, it doesn't matter if you're getting this year's flu vaccine or last year's flu vaccine. Those vaccines, those strains, we know from decades of doing flu vaccines that they're not really going to be different from one year to the next in terms of the safety profile. So we know there are risks for the flu vaccine. The flu vaccine is grown in chicken eggs, and that's the best way to grow large quantities of influenza viruses at scale for manufacturing it. So obviously people with egg allergies can't take the flu shot. We've been doing flu shots, inactivated flu shots, for so long that it doesn't really matter. We know that it doesn't really matter what strain it is. The biggest risk of the flu shot is that we chose the wrong strains, and they're not going to be protective for the actual influenza strains that are circulating. And that is one problem with the flu shot. We're making a best guess every year which flu strains are going to be dominant that we need to be protected against, and that we don't always get that right.
You mentioned at one point about antibodies, and the idea that there are these antibodies in your blood that are essentially kind of optimized for a particular shape. They're a glove optimized for a particular shape, a particular pathogen. So is that one way that you can do testing if someone has been exposed to a virus? And then what other ways can you do testing? And
yeah, absolutely. So there's really 2 kinds of tests that we talk about when we talk about testing. One is called serological testing, which is for antibodies. Essentially, it's what you just described. That can tell you if you've been infected before. It doesn't always tell you if you're infected now. So one complicated thing about antibodies is that there are a few different kinds. So there's IgG. Those are usually what we think of when we think of the neutralizing antibodies. But there's another type of antibody called IgM, and those are the antibodies that you have that are existing at low levels in your body all the time. IgM antibodies are great for triggering immune responses, and they have some ability to inactivate the virus, but their main role is to recognize that the virus is there. IgM antibodies start going up a few days after you're infected, up to a week after you're infected. IgG antibodies take longer to increase. So if you have an antibody test that only measures IgG, that's probably going to be looking at people who had been infected before but are no longer infected now. If you have a test that is looking for IgM and IgG antibodies, which some of the tests are, you may be able to identify people who are actively infected at that time by detecting IgM antibodies. But it's really, you wouldn't be able to detect every infection. And so that's why serological tests are great for surveilling prevalence in the population. They're great for being able to tell if you had COVID and you couldn't get a test because you weren't sick enough, but you want to know if you had it. It's excellent for that. It's excellent for informing epidemiology studies and trying to understand how many people might have immunity, where we go from here. But they're not great for doing diagnostics of people who are infected right now. For that, we use a different type of test called PCR, and PCR looks for the virus's genetic material very specifically right then. So you don't have that genetic material in you if you've cleared the infection. Now, there have been these reports of reinfection, and increasingly the data looks more and more like that is some residual viral RNA, the genetic material in patients who've recovered that aren't actually producing any new virus. So it's just basically the leftovers from their infections. But when people are symptomatic and going in, that's when your virus loads are highest. So when you go to the clinic and you need a test, the PCR test can detect that virus. So it can detect you when you're infected. It can also detect, one thing we've learned about COVID in the last couple months is that people can transmit the virus before they become symptomatic. So that's probably why this has been a problem with community transmission, because people are spreading the virus before they even realize that they're sick. And so the PCR test is sensitive enough that it can detect virus before people become symptomatic, which is why it's better than an antibody test for actually identifying people who are infected.
Are there any other issues, misinformation, misunderstanding that you've seen going around that you want to clear up, things that you think are not maybe coming through the news or media outlets?
Yeah, so every week there's something different. And usually, I think in closing, I'd like to say that, I mean, one thing about this pandemic that has been really encouraging to me is how the scientific community has really come together and communicated with each other and collaborated with each other in great ways. And one way is the availability of data so quickly. One of the problems with this, however, is that it's often covered by the media in a sort of inaccurate or sensationalized way. And I think some of this is just, as a society, you know, we're really, the news cycle is being dominated right now by this. And we are really desperate, going stir crazy in all of our houses, hoping for some news that will give us some clarity about the future and even the immediate future. These are really strange times that we're living in, and it feels like time is going by really fast, but at the same time, it feels like an eternity. I can't believe March was only 2 months ago. It feels like a year ago. And so sometimes when a finding that would normally be incremental or not that exciting is treated as like some sort of earth-shattering breakthrough by the media. And oftentimes these are things that can really scare people. For example, yesterday the LA Times reported on a new paper that suggested that a variant of SARS coronavirus 2 that is dominant in the United States and in Europe is more transmissible. And that's not really what that study showed. It offered that as a hypothesis, which of course needs to be tested. but that distinction gets lost in the reporting sometimes. And so the net result is that people get really scared. Oh my God, SARS coronavirus 2 is mutating to become more transmissible. We're never going to get rid of this thing. This is the end of civilization as we know it. And I'd just like to encourage people that most of the time when you see stories like that in the news, they're not the whole story. And I just encourage people, as well as reporters, to talk to scientists, not just virologists like me, but immunologists, epidemiologists, people from all disciplines, bioengineers, mechanical engineers to some degree. Certainly, there's a lot of mechanical engineers involved in testing equipment and testing capacity, as well as electrical engineers. So there's a part for a lot of different disciplines to play. And I just really hope that people take media reports with a grain of salt, because oftentimes they're really just scratching the surface of a much larger story, and look to the scientists to help put it in perspective.